Hello students, welcome back to the science class. In this video, we are going to learn about the structures of kidney and the process of how urine is produced. So without further ado, let's begin our lesson. The biological process involved in the removal of harmful metabolic wastes from the body is called excretion. As in other processes like nutrition, respiration, transportation, different organisms use different strategies to do excretion. In case of unicellular organisms, wastes are removed from the body surface into the surrounding water by the process called diffusion. But in case of multicellular organisms, specialized organs are evolved to perform excretion. Excretion in human beings. The chief organs that is involved in excretion is kidneys. The excretory systems of human beings consist of first, a pair of kidneys, right kidney and left kidney. Second, a pair of ureters, right ureter and left ureter. Third, a urinary bladder. Fourth, a urethra. Kidneys are located in the abdomen on either side of the backbone. Kidney is the basic filtration unit. It consists of a very thin-walled blood capillaries. Each capillary cluster is associated with a cup-shaped end of a tube that it collects filtered urine. Each kidney has a large number of filtration units called nephrons packed close together. The waste products are nitrogenous wastes that are collected from the blood in the form of urine. The produced urine passes through the ureters into the urinary bladder. The urine is stored in the urinary bladder, then the urine is released to the outside through urethra. Now, let us learn about the structures of the nephron and how urine is produced. Nephron is the basic functional unit of the kidney that consists of the renal corpuscle and the tubular attached to it. The word nephron is derived from the Greek word nephros, meaning kidney. Each kidney contains approximately 1 million nephrons, which perform several important functions of the body, including waste excretion, maintaining the body's pH, regulation of blood pressure, and several other functions. So let's go through the structures and the function of the nephron and learn more in detail about the different parts of the nephron and the role of the renal tubules in the urine formation and excretions process. Let's first start with the basic structures of the nephron. So, nephron are microscopic structures located in the kidney, and kidney has two important growth regions, an outer cortex and an inner medulla. If you look at this nephron, you can see that the part of this nephron is present in the cortex of the kidney, and the part of this nephron is placed in the medulla of the kidney. This is an important point to remember as it plays a significant role in the formation of the urine that we will discuss later in this video. So, the nephron is a tube-like structure whose length varies from 35 to 55 millimeters, and the structures of the nephron can be basically divided into two major portions, the Bowman capsule and the renal tubule. At one end, the tube is close-folded and expanded into a cup-like structure, which is known as the Bowman's capsule. Now, the Bowman capsule that was discovered by this guy, Sir William Bowman, at the age of 25. He was an English surgeon and histologist who is also credited for the publications of the two historical books, The Physiological Anatomy and Physiology of the Man, Encyclopedia of Anatomy and Physiology. So, coming back to the nephron, we discussed about the cup-shaped structures at one end, which is known as the Bowman's capsule. The space inside the Bowman capsule is known as the Bowman's space. The Bowman capsule encloses a cluster of blood vessels, which is known as the glomerulus. The word glomerulus is derived from the Latin word glomus, meaning the ball of the thread. So, we can see why this is known as the glomerulus. The blood vessels which brings the blood inside the glomerulus is known as the afferent vessel. And the blood vessel taking the blood out of the glomerulus is known as the efferent vessel. Remember, A comes before E, so afferent brings the blood in, then efferent takes the blood out. The Bowman capsule and the glomerulus together constitute the renal corpuscle. Okay, so this was about one end of the nephron. The other end is known as the renal tubule. The word tubule means a small tube. As you can see, the renal tubule is a long and folded tube-like structure that emerges from the Bowman capsule at one end and it ends into the duct systems of the kidney. The tubule is divided into three main parts based on its function. The proximal convoluted tubule, which is a part of the tubule shown in the light blue color. Then you have the loop of Henle, which is shown in the dark blue color. And finally, we have the distal convoluted tubules, which is the part of the tubule here shown in the orange color. The distal convoluted tubules in turns ends into the collecting duct. So the first part of the tubule is known as the proximal convoluted tubule. Proximal because it is close to the Bowman capsule, and convoluted means it is highly folded structure that helps to increase the overall length of the tubule in a small space. The proximal tubule is located in the cortex of the kidney. The proximal tubule leads to the loop of Henle, which is the second main part of the renal tubules. This region forms a loop that is located in the renal medulla. The loop has a descending limb and ascending limb. The ascending limb of the loop Henle leads into the distal convoluted tubule. The distal means away since it is located far from the Bowman capsule as compared to the proximal tubules, and convoluted since it is also folded structures to increase the overall length of the tubule. The distal convoluted tubule drains into the collecting duct, which also receives input from many other nephrons in the region. The collecting duct drains into a larger collecting duct and ultimately into the renal pelvis, which is connected to the ureter. The ureter drains the kidney into the bladder. The distal convoluted tubule is also located in the cortex of the kidney. Along the wall length of the nephron run this network of small blood vessels known as capillaries that brings in pure blood containing many waste metabolic products and takes out pure blood that has been filtered by the nephron. The impure blood or the arterial blood comes into the kidney by the renal artery, and the pure blood, which has been filtered of all metabolic and other waste product, goes back to the body by other renal veins. So this was about the structures of the nephron. Next, let's have a look at its basic function. So nephron uses four main mechanisms to convert blood into urine, which are filtration, reabsorption, secretion, and excretion. In this video, we will understand the mechanisms of urine formation in a better way. We will look at how the blood is filtered at the level of the renal corpuscle, and then we will understand how your urine moves forward in the renal tubules and how different mechanisms like the absorptions and secretion apply in different parts of the renal tubules. So, blood enters the kidney via the renal artery. This is a large artery then branch of aorta. After entering the kidney, the renal artery gets divided into small divisions until it reaches the smallest one that enters the nephron. This is the afferent vessels that brings the blood into the glomerulus, and this is the blood vessel that takes the blood out of the glomerulus, and this is known as the efferent blood vessel. In between these afferent and efferent blood vessels is a jumble of mass of capillaries that is known as the glomerulus that we discussed earlier. Glomerulus is derived from the word glomus, which means a ball of thread. The glomerulus is a tuft of blood vessels that is located inside the Bowman capsule. This tuft of vessels greatly increases the surface area for filtration that comes in contact with the Bowman capsule, and this is where the mechanisms of urine formation takes place that is known as the filtration. Understand the filtrations at their site, you have to know a little bit about the glomerular filtration barrier. So if you take this part of the renal corpuscle and look at this microscopic view, it will look something like this. This is the wall of the blood vessel. Outside, this is the basement membrane of the glomerulus, which is in contact with the wall of the Bowman capsule. The wall of the Bowman capsule contains special type of cells which are known as the podocytes, and between these podocytes we have a small gaps, as you can see, and these are known as the filtration slits. Together, these three layers form the glomerular filtration barrier that acts like a filter paper. So we have the blood coming in this blood vessel that gets filtered through this glomerular filtration barrier to form the impure plasma that is passed down into the renal tubule. The glomerular blood pressure provides the driving force for the water and solids to be filtered out of the blood plasma and into the interior of the Bowman capsule called the Bowman space. Only 1/5 of the plasma is filtered in the glomerulus and the rest passes into the efferent arteriole. The things that do not get filtered are things like the red blood cells or larger molecules like proteins. Also, one important thing that you should note here is the difference in the diameter of the afferent and efferent blood vessel. As you can see, the efferent vessel is of the smaller diameter as compared to the afferent vessel, which means that the blood is coming in through a vessel of larger diameter and leaving through a vessel of smaller diameter. This increases the pressure inside the glomerulus, which provides a driving force which makes this filtration possible. The kidneys can adjust the rate of the glomerular filtration by adjusting the pressure inside the glomerulus, and this is all possible because of the diameter of afferent and efferent blood vessels can be adjusted. The plasma that gets filtered into the Bowman space is known as the glomerular filtrate. The glomerular filtrate next moves into the renal tubules where it is further processed to form urine. This further processing is very important as the initial glomerular fluid that is filtered into the Bowman space contains a lot of things that shouldn't be excreted out with the urine like the sodium chloride, glucose, amino acids, and excess water. The first part of the renal tubule is the proximal convoluted tubule where approximately 2/3 of the filtered salts and water, almost all the glucose and amino acids are reabsorbed. We don't want to lose glucose, that's hard-earned stuff that we ate that was good for energy. We don't want to lose necessarily as much sodium chloride as well. This is achieved by the method of reabsorption. The wall length of the renal tubule is surrounded with the blood vessels which are known as the peritubular capillaries. Whatever that gets reabsorbed is then passed back into the blood circulation. This is known as reabsorption and not absorption because the same nutrients have been absorbed once from your gut into your blood by the similar mechanism. The wall of renal tubules has many types of protein channels that reabsorb some of the important things that shouldn't be excreted with urine. No, but 70% of the sodium chloride gets reabsorbed in the proximal convoluted tubules, and same 70% water gets reabsorbed here, and more than 95% of the glucose and amino acids get reabsorbed in the proximal convoluted tubule. Next, the filtrate passes into the loop of Henle. The loop of Henle has some role in reabsorption of important salts, like about 25% of the sodiums get reabsorbed here. Similarly, 25% of calcium, 20% of potassium, and about 60% of magnesium get reabsorbed in the loop of Henle. But the loop of Henle has a major role in the concentration of urine, and this is a complex topic and usually taught at the level of the med school physiology, but I'll try to simplify this as much as possible, so pay close attention. So here is our sections of the kidney, and you can see how the structures of the kidney is divided into an outer cortex and an inner medulla. The inner medulla of the kidney is very important region as this contains higher concentrations of salts compared to the cortex of the kidney. This higher concentration of salts in the medulla is made possible by the loop of Henle of the nephrons. So here is our nephron inside the kidney, and you can see how this loop of Henle is present inside the medulla of the kidney and the rest of the nephron is present up in the outer cortex. The loop of Henle has a descending limb and ascending limb. So if you look at the ascending limb of the loop of Henle, it contains special ion channels that pump out ions like sodium and chloride, thus making the renal medulla more salty or hypertonic. The ascending limb, however, does not allow water to move out as it has no channels in its wall that allow the water to move out. These types of channels that allow water to move out are known as the aquaporins, and these types of channels are usually present in the descending limb of the loop of Henle that makes it freely permeable to water. So now this is all salty and the water will move out of the descending slim into the renal medulla and then eventually into the blood vessels and get reabsorbed, and this is the reasons why loop of Henle is so long. That is to give the time for this water to secrete out, and that's why it dips nice and pretty far down into the salty portion, which is the renal medulla. So this is the major mechanisms of gaining back a lot of waters that gets filtered up at the level of renal corpuscle. So after the loop of Henle, the water then passes into the distal convoluted tubules, which is this part of the renal tubules, and here we have more reabsorptions of ions like calcium and sodium. We are just reabsorbing more things that we didn't want to lose in the first place. There's a lot of things we could talk about what gets reabsorbed here, but this is just the overview in the distal convoluted tubules. The final mechanism of urine formation also takes place. This is known as secretion. You can view the secretion as the opposite of the reabsorption. In secretion, the waste ions and hydrogen ions pass from the capillaries into the renal tubules. This is the final step in the formation of urine, during which wall of the tubule will actively remove the waste substances which are harmful to the body from the blood that have escaped the filtration. The secreted ions combine with the remaining filtrate and become urine. The major substances secreted into the distal convoluted tubules include hydrogen ions, potassium ions, and ammonium ions. The mechanism secretion also takes place in the proximal convoluted tubules, but to a lesser extent, and some important stuff that gets secreted into the proximal convoluted tubules include substances like urea, uric acid, creatine, ammonium ions, and some other drugs. Then at the end right here, we have our filter that has been processed. A lot of water has been taken out. It's a lot more concentrated. We have reabsorbed a lot of salts, electrolytes, and we have reabsorbed all the glucose and amino acid that we cooked. This waste fluid then gets dumped into the collecting duct. You can see the collecting ducts like a common drain pipe that collects waste from all the houses in the neighborhood, except in this case, where this pipe is the collecting duct and the houses are individual nephrons. The collecting duct is important functionally for two aspects. First, it dips back into the medulla, and second, because antidiuretic hormone acts on our collecting ducts to decide whether our urine should be concentrated or diluted. The function of antidiuretic hormone is a whole different topic that should be discussed in another video. The urine in each collecting duct is drained into the renal valves, which is this dilated portion of the kidney. This is in turn connected to the ureters. The ureters connected the kidney to the urinobladder that is connected to the urethra. I think that's all about our lesson today. Don't forget to do the exercise on Edmodo. Bye!